All right, hi everyone. Thanks for sticking around. I'm talking to you from Ghana country and pay my respects to the Ghana people and extend that to the First Nations people all over Australia. I would like to share with you a project that is very dear to my heart. It combines my passions of working with people and working with insects and raising the awareness of taxonomy in the general public. And it's a project called Insect Investigators. And it's all about bringing together scientists and schools to help discover and document Australia's biodiversity. So for those of you I don't know, I'm a postdoctoral researcher at the University of Adelaide. I'm currently on an ABRS postdoc fellowship. I'm also an honorary researcher at the South Australian Museum, and my expertise is in parasitic wasps. So I work on a family called Braconidae, specifically on a subfamily called the Microgastrinae. They're only a couple of millimeters long. You've probably heard me talk about them at previous AES and SASB conferences. They're parasitoids of caterpillars. They do some really cool stuff in the environment. But Insect Investigators is about school kids, and it's about getting them involved in how we actually go about documenting the insect life around us. And it's about really trying to get taxonomy out there in the general public. You know, my dream is that if you ask a school kid what taxonomy is, they would be able to tell you, it's discovering and naming species in the same way that if you ask a school kid what astronomy or paleontology is, they're often able to explain what the word means. Now, I'm preaching to the converted, of course, but as we all know, only a very small portion of Australia's insects are described. And I just want to make it clear that I'm not trying to suggest at all that citizen science can solve this issue. The only way we're going to document Australia's biodiversity is with better funding for taxonomists and more boots on the ground of professional, employed, trained taxonomists. But I also believe that to be able to get that increased funding for taxonomy, we need the general public on our side. And we need people to be as excited and passionate about species discovery as we all are. The other aims of the project are about building the DNA barcode library for Australian insects and trying to get us up there with some of those countries who are much faster and better at getting the DNA barcode libraries done for their fauna. And it's about describing new species actually in collaboration with communities. So, bringing a small portion of that really important work that we do every day in describing species into the general public's, living room, so that they're actually part of that process, and hopefully then feel more connected to it and support us all having jobs in the future. So what I'm going to do is talk to you about the initial trial of this project, and then talk to you about the current exciting expanded version that's underway. So the initial trial happened over 2019 and 2020. I worked with 4 regional schools in South Australia. So Cow area schools over on the Eyre Peninsula, about 5 hours drive from Adelaide. Waikerie and Ramco Primary School are in the Riverland of Australia, and Macclesfield is in the Adelaide Hills. And all of these schools ran a malaise trap for me. So for the non-entomologists, a malaise trap kind of looks like a tent. It's a passive trap that flying insects like wasps and flies fly into. They hit that vertical wall in the trap and then instinctively head up to the top of the malaise trap into a collecting bottle, which can have a preserving liquid like ethanol or propylene glycol in it. So as part of the project, I headed out to each of the 4 schools. I ran a workshop about insects and taxonomy and biodiversity, and we set up a malaise trap together. I then left them with the malaise trap. Some of the schools only for a few weeks. For some of the schools, it was there for several months. And they each sent me in the samples back here at the University of Adelaide. And we then sorted them. So we sorted each of the samples to order. Being a hymenopterist, I was most interested in the wasps, although I did pass the flies on to Keith Bayless over at ANIC, and he found some cool things as well. And all of the schools actually found some microgastrines, so the group that I am working on during my postdoc. And all through this process, I wrote blogs and I recorded little videos of what was happening in the lab so that the schools could know what was going on with their sample after it got back here into the lab. And all 4 of those schools actually found new species of the group that I'm working on. So I went back out to each of them and we ran a second workshop about, how do we name species when we find them, and how does the process of describing a species and making it a formalized entity actually occur. And all 4 of the schools got to name the species name for their new species. So we've got Franklin harborensis, which is named after Franklin Harbour, the council area that Cow Area schools in. And we've got Dryoplanitis, which means bush wanderer, which the Macclesfield school called their wasp. We've got Rhamco marmorata, which combines the Rhamco Primary School's name with the Latin word for marbled, because they thought the wasp looked like polished marble, and Wakery Yetis. So Yetis is the youth environment team's nickname, and they were the group at Wakery Primary School who were running the project. So it's really cool to work with the kids and come up with ideas for species names, and maybe steer them towards some that were a little bit more appropriate to be published. So we published all 4 of the new species in an article in Zoo Taxa that's open access, so you can go and check it out if you want. And it was really important to me that we somehow acknowledged that the students were actually authors of this project as well. So we included them as an author on the paper. And whenever you're working with community, I think it's really important to really acknowledge their contribution in a formal way. So this is just a word cloud. We asked the students at the end of the project what was their favorite part of being part of the insect investigators. And the larger the word, the more common it was that it came up in their responses. So finding the new species, finding the new wasp was sort of the favorite things. You'll see lollies is quite a large word. We did an activity where they had to classify lollies and build phylogenetic trees using lollies. And then I did the survey straight after that activity. So I learned that it's important when evaluating to put a little bit of distance between some of the activities. We also asked the students to self-evaluate what they felt they had got out of the project. So for example, we asked the question, did your interest in insects either increase, decrease, or did it stay the same as a result of the project? So dark blue is it increased a lot, light blue is it increased a bit, grey is it stayed the same, orange is it decreased a bit, and red is it decreased a lot. So blue is good, grey is neutral, orange and red is not so great. So way more than half of the students thought that as part of the project, their interest in insects increased. Nearly all of the students felt that their knowledge about insects in the local area increased. About half felt their desire to protect the environment in their local area increased, which was a really great outcome. About half felt that their connection to nature increased. Slightly more than half felt their desire to spend time outdoors in nature increased. And about a third felt their interest in pursuing a career in science increased. For a lot of students, their interest stayed the same. And for a few students, they felt that their interest in pursuing a career in science actually decreased. And this, I think, is a really interesting outcome and something I wish I'd had time to delve into a little bit more, just to find out whether that had to do with the type of science we were pursuing as part of the project, or whether it had to do with the fact they learned science takes a really long time. From the time I came out to them and set up the Malay strap to the time the species were published was often more than a year, and whether that realization of that science can be tedious and long influences students' decisions, I'm not sure. But overall, we had some really good outcomes, which led me to pull together the most amazing team ever, who I will introduce to you soon, to apply for a citizen science grant that was funded by the Australian government's citizen science program. So these are fairly large grants that are specifically for citizen science and for trying to connect the community to the scientific process. So that logo banner there are all the wonderful organisations that are part of that grant. We were very lucky to be successful and get funding for this project. So it's being led by the South Australian Museum, but you can see that there's a whole bunch of really incredible organisations involved. So this version of the project is going to involve more than just 4 schools. We are looking at, or we are working with 50 schools that are across regional South Australia, Queensland and Western Australia. We're also going to try and involve some of the landcare groups who are located near the participating schools to try and help build some more sustainable connections in these regional communities between the schools and the people who are there on the ground, helping preserve the local environment and working in it. It's also not just going to be me looking at microgastrines, so trying to encourage the involvement of lots of taxonomists with expertise in different groups. So the project will work something like this. In our lab at the moment is sitting 50 Malay straps in boxes that Alana and I are getting ready to package and send out for next year. So each of the schools is going to set up a Malay strap in an area of native vegetation on or near their schoolyard. That Malay strap's going to run for 4 weeks in term 1. So during the summer of next year, we're going to be running propylene glycol, and each week the students will take the bottle off full of our insect soup, send it into us here and put the fresh bottle on. So that'll go for 4 weeks. Once we get all the insects here, we're going to be sorting them and picking out our target taxa for DNA barcoding. So this will be guided by the taxonomists who are involved in what groups they're really interested in looking at from the schools and trying to get a bit of a representative sample. We're going to be DNA barcoding 3 plates of samples, so nearly 300 specimens from each of the schools. And that's working with the University of Guelph, so the home of Bold in Canada. We get the specimens back from Bold, we'll send them out to all our wonderful taxonomists who will be able to tell us a little bit more about them and who may even be able to say, look, here's a species that I can tell straight away is new because it's a group I'm working on. And in that case, we will head back to the schools, do some more workshops on taxonomy and actually get the school to work with that taxonomist to help name that new species. So there is an amazing group of people involved. Very quickly, just to acknowledge them. Andy Howe, based up at University of Sunshine Coast in Queensland, looking after the Queensland part of the project and also the ecological component of it. Alana is looking after all our logistics and equipment. Brock's running our social media and website. Andy Austin and Steve Denellen are both providing some incredible advice on the project. Sylvia and Alice are looking after the South Australian schools. Karen Cullen over in WA is looking after the WA schools with Nathan Ducker at the WA Good League, and Leanne at the museum, and Kate from the South Australian Science Teachers Association is helping us make some really great educational resources for schools and teachers to go alongside. And obviously a project like this cannot happen without our amazing museum staff. So a big shout out to Ben, Matt, Chris, Susan, Nick and Mark, who are helping make sure that all the specimens we collect during the project end up in museums and provide a great resource into the future. So to finish up, these are the 50 schools that we are going to be working with next term. There's some amazing locations, a really incredible spread of samples that we're going to be having in. So if you are a taxonomist who works on things that come into Malay straps and you want to be involved in the project, you can find more information on our website, follow us on socials, and I cannot wait next year to hopefully present some results from the study.
Fantastic. Thanks, Erinn. Are there any questions in the chat or for Erinn, or just unmute yourself because we're at the end and we can all just like chat now if you want to. Quite a few comments in there, Erinn, about the names being awesome. The word, the words in that like word thing that lollies was, the second biggest word in that.
Yeah, I learned a lot about social science and that it's very important about when you conduct your evaluation surveys.
Yeah.
And speaking of evaluation, I think there's a question in the chat from Lynn about are we going to do a before response and an after response? We did try this with the initial trial and it was really difficult to try and tease apart what's going on, and really to do it properly we would need to have control schools who are not part of the project and also survey them and who are doing the same curriculum, and yes, very difficult. simple answer is we've decided not to do pre-surveys. We're going to do post-surveys, but also a lot more qualitative evaluation. So we're going to be running some photo story evaluation and some focus groups to try and get a bit more of the qualitative stories out of outcomes out rather than the statistics, which because I don't really know how much they actually show us.
Another great question here from Catherine. What age group are the school kids?
Yeah, good question, because it was a hard decision on our behalf as well. And there was a lot of discussion about whether we restricted it and tried to target specific groups, but we actually left it really open and just saw what schools applied. Something I didn't mention was that we had nearly 200 schools apply across Australia, and we were only able to work with 50. And the 50 we're working with range from junior primary right up to year 11 students. So across the whole spectrum.
Erinn, there's another question.
Thank you.
No worries. Erinn, in your project, will you collect candidates of new names from school kids and provide to taxonomists in case they have nothing to name whenever they find a new species?
I think it's important that the school kids are involved in naming a species that's been collected in their local area rather than, just anything a taxonomist finds in the future, because it's all about trying to build connections between people and the space that they're currently in. What I would like to do is make sure we maintain the connections for a long term, because as we know, taxonomic processes can be very long. So what I would love is if in 5 years someone's using a specimen that was collected as part of the insect investigators, that they're able to trace that back to our team, who can put them in contact with the school if they did want to go back and work with the school to help name it.
Cool. Does the school get to keep any of the insects they collect as a reference? That's something we've deliberated about, hasn't it?
Not at this stage, no. The plan is for them all to be properly databased and registered and put into museum collections. Bold will be imaging the ones that are sequenced as part of their pipeline for barcoding. So those images will be available on Bold for the schools to be able to see, but no, not planning on sending back the specimens, but something we can maybe talk about, especially for those groups where we've got, lots of representatives of single morpho species. It might be a nice idea to be able to provide schools back a little display kit of, here are some of the really common insects from your area. That's a great idea.
Okay.
Erinn, you should talk about the problems in trying to Latinize some of the new species names that the schools want to run with. I remember that being sort of, oh my God, where do we even start here?
Yeah, poor Mark is my go-to person for trying to make sure names apply to the code. Yeah, look, it's an interesting process working with schools to come up with names. We had to steer away from some ideas that are, wanting to call them a big stingum or something after the big ovipositor and all sorts of things. So yeah, it's definitely a process of first getting the idea to be something that the whole class can agree on and that kind of makes a little bit of sense, and then turning that into a name that's going to agree with the code.
I love Ying's idea here. Maybe we could get 3D scans of them and print one for the schools, which would be such a cool thing to send them. See how much of the budget is left for 3D printing.